Okay, so before we talk about DNA replication, I just want to go over the basic structure of DNA. So DNA is a double helix. The base pairs, that is the interior portion of the DNA double helix. Remember, apple in the tree, car in the garage. A with T, C with G. So A is adenine, T is thymine, G is guanine, C is cytosine. The backbone of DNA, we call it they call it a sugar phosphate backbone or a phosphate deoxyribose backbone. The sugar in DNA is deoxyribose. That means it's a ribose sugar that is missing the hydroxyl group, the OH group there. So that missing OH group is why it's deoxyribose. Ribose sugar, what's in RNA, would have an OH group there. And then the phosphate, so then there's the phosphate portion of it. Okay. Anybody remember what kind of bonds hold together the base pairs? Is that hydrogen? Yeah. Yep. Hydrogen bonds. And why? What's the benefit of hydrogen bonding being between them, Thomas? Yeah, they're easy to break, so you can, because we're going to have to break them to do DNA replication, and you need to break it to use the DNA. But they're also strong enough so that it doesn't just fall apart. Okay. And then directionality. It goes. It's anti-parallel, meaning parallel, like the lines don't intersect, but anti, they're going opposite direction. It's like a highway, right? Where one goes 5' to 3', the other goes the opposite direction, 5' to 3' that way. Okay. Understanding this idea of 5' and 3'. Oh, and understanding that 5' is phosphate, and 3' is the hydroxyl, the OH. That's something. So 5' equals the phosphate end, and 3' is the OH or the hydroxyl group. It's a little bit of trivia, but it's just something they would want you to know. All right, getting into the actual section then. So if we look at DNA, complementary, meaning complementary, like as in, like two puzzle pieces go in together. They're not identical, but they complement one another. They're opposites of each other. So the A pairs up with the T, the C pairs up with the G. And when they discovered that it had these base pairing kind of rules, it brought up a kind of an obvious system for copying the DNA. It's like, oh, if you split the DNA, this strand of DNA and that strand of DNA, they maintain the same amount of information. In other words, by splitting this DNA, you don't actually lose information because you know that a T has to go there and a G has to go there. And that's kind of interesting because normally if you had a book and you start removing letters, you do actually lose information. But DNA, because it's complementary base pairing, by splitting these DNA strands, you can then copy each strand and double the amount of information, or not double the amount of information, but double, you have two copies of it now, but you don't lose information by separating them. Okay. And that's what they mean by the structure versus function. Remember the scientists, you don't have to know the scientists for the AP exam. I think it's interesting. It's something you learned a lot about in a lot of your other biology classes. That's what 13.1 and the first part of 13.2 is on. Don't even know the experiments and stuff, although they are interesting. So here, each strand stores the information to reconstruct the other. There you go. Interesting thing with copying DNA, it is very fast and very accurate. So the cell can do it pretty quickly, and it'll do it without much error. I'll show you how the errors come into place and what we do with them a little bit later on in the section. You need to know the proteins and the enzymes that are involved in DNA replication. The AP exam isn't necessarily going to have you draw the whole thing out, but they will want you to know what these different kind of proteins do. And how we don't really know exactly how it works in human cells, DNA replication. We really know how it works very well in bacteria, specifically E. coli, but we do know that the how it works in bacteria is very similar to eukaryotic cells. Okay. Anyways, how it would work in an E. coli cell. Remember that bacteria have one circular chromosome. So if a bacterial cell is going to copy its DNA, they would, and you have this in eukaryotic cells, an origin of replication. So it's exactly what it sounds like. The origin of replication is where we start replicating the DNA. So you would pull apart the DNA, those double strands there, and then you start copying this way and that way. So you start seeing, they call it the replication bubble because it looks like a bubble forming, where the bubble will go this way and that way, and it'll keep going until you form two new daughter cells. Okay. John, honey? Yep.
You're gonna have an external background.
I do, yeah. Right here. So there you go. There isn't really a whole lot more than that on this picture. Obviously, it's more complicated. We'll get into like the actual enzymes and proteins involved, but that's the idea of how it works in E. coli cell. Human cells, we have multiple origins of replication, so that's a big difference to understand. Bacteria cells is just one origin of replication. Human cells, eukaryotic cells, they all have multiple origins because we have all these different chromosomes, this long linear chromosome. All along that linear chromosome, we're going to have these replication bubbles. Okay, so here's a bubble, here's an origin of replication, there's one, and then we start, once we get the bubble, just like you saw in the prokaryotic cell, you'll go on either side of the bubble, and these bubbles end up meeting each other, and then once all the bubbles have met each other, we're then done replicating that specific chromosome. Okay? All right. Let me make sure I set everything. And the purpose of multiple bubbles is it speeds up copying. Kind of obvious. All right, so now getting into the proteins and stuff, the details of it. So the thing with DNA polymerase, specifically DNA polymerase 3, not 1, 3, is what is the main actor in copying DNA. Now, DNA polymerase, it can't, what's interesting about it is it can't just start, you could open up the DNA, but DNA polymerase 3 can't just start adding the new nucleotides to make the copy of it. DNA polymerase needs what's called a primer. Like if you ever have painted a wall, like there's something called a primer layer you put down. It helps like the actual paint stick to the wall. That's why they use the word primer here. It is an RNA primer because you'll have this enzyme called primase. ACE, that's the ending that signifies enzyme. Primase puts down this RNA primer. Now notice the direction. You have to know the direction of these things. DNA polymerase goes from 5' to 3'. Remember 5', that's the phosphate end of the backbone. 3', that's the hydroxyl end, OHN. DNA polymerase, think of it as it goes downhill. 5' to 3', you're counting down. DNA polymerase goes downhill. So that's why the direction goes this way. So you have this RNA primer, and then DNA polymerase can come in, and you'll see it on the next slide. It'll start adding the nucleotides right here, and then it'll start going all the way down. Now, another thing that's happening is we have to keep unzipping the DNA, almost like you would a zipper. The enzyme that unzips it is something called helicase. And I don't know, that always made sense to me, the name, in a way. Like, it's the helix, and it's like breaking it open. So helicase unwinds it. And then there are these things called single-strand binding proteins. SSRB, you could think of them. These single-strand binding proteins, they bind to the single strands, and their job is to keep the strands open. And that makes sense, right? You would need something to prevent these two DNA strands from coming back together. Okay? There's also something called topoisomerase, and this is kind of a goofy idea here, but think about it. And you actually see it with like like the cord for the camera. Like, what do you notice about my camera cord? It's really, really tangled, right? So the issue is helicase. If you keep unwinding the DNA, it's going to start knotting like that the cord of the camera will. Topoisomerase, its job is to reduce the tension. It'll basically, as helicase is unwinding the DNA, topoisomerase will prevent those like knots and kinks from happening by breaking the DNA and unswiveling it ahead of the helicase. Okay? So there you go. Those are the main proteins involved there. Replication fork, like a fork in the road. So the replication fork is, it goes in the direction that we're copying DNA. And then we copy DNA 5' to 3'. All right. Make sure I set everything here. Topoisomerase. There you go. Okay. The length of the primer is about 5 to 10 nucleotides long. Not super important, you know that name, but understand it's made of RNA, and it's just a short. The purpose of it is just to get DNA polymerase going, because DNA polymerase can't add nucleotides just by itself. DNA polymerase, when they say it adds nucleotides to the 3' end, that's because it's going 5' to 3'. So it'll start adding those nucleotides at that OH end of the next nucleotide in that chain. And it's kind of interesting just how quickly it can happen. In bacteria, 500 nucleotides per second. That's really freaking fast. It's a lot slower in eukaryotic cells. It's only 50 per second. But I don't know, just to give you an idea of how quickly this stuff happens. It's very, very fast. Very efficient. Okay. Looking at a little bit of the mechanics of how the bases get added in. The bases, when they come in, you will have 3 phosphates. Now, normally, whenever the bases are there, there's only 1 phosphate. But if you look at this, and let's say I change this to A, what does that remind you of? The structure of that. ATP. Yeah, exactly. This is essentially ATP. Now, with the exception that ATP had a ribose sugar. This is a deoxyribose sugar because it's missing that OH group there. Anyways, we bring in that nucleotide. In this case, it's a T. It's thymine because it's pairing up with an adenine. And we'll end up removing the 2 ending phosphates. They call that pyrophosphate. Now, this reaction here is an endergonic reaction. If you remember those kind of words. Endergonic meaning it requires energy. Think of it's like a ball going uphill. But then this pyrophosphate, what this reaction is showing here is we will then break those 2 phosphate bonds. That is an exergonic reaction, meaning it releases energy. We can then couple those 2 reactions, meaning the energy released by breaking the pyrophosphate, we can then use that to fuel adding the G of the next nucleotide. So it's kind of a clever system that the body has going on there. Okay. And make sure I set everything here. Any questions so far? All right, so let's get into the details of it. And this would be especially something where reading before class would have been really helpful. It can feel like a lot all at once, but just do the best you can to wrap your mind around this. And keep in mind that a lot of the questions you're going to see on this is them asking, what does this protein do? What does that protein do? Okay? All right. So here's our bigger picture. We have the origin of replication where we break open the DNA and you get that replication bubble. Now, we copy DNA 5' to 3'. Why this looks so goofy is because it can only go 5'. Think of it as a one-way street. You can only go on like this way on a one-way street. It's kind of like if you ever had to get home on one-way streets, you have to end up doing like a box to get back around. This is kind of what can happen with leading strand and lagging strand of DNA. Where when you break open the DNA, Here, going this direction, that is going, this would be 5 prime, that would be 3 prime. So we start copying DNA 5 prime to 3 prime. Like we can just put the primer down, DNA polymerase can just start going. Call that the leading strand. You'll get another leading strand here. This would be 5 prime, that would be 3 prime. DNA polymerase can get going. But thinking back to like one-way streets, an issue happens is, well, what about the other side of each of those origins of replications? How do you copy that side if DNA polymerase can only go 5 prime to 3 prime? Well, basically you have to do them in fragments. We call these Okazaki fragments. Yeah, the scientist discovered his name, last name was Okazaki. That's the. So basically, we have to synthesize them in these little fragments. I'll get into the details of it in a couple slides, but that's what's going on there. It's basically just like one-way streets. It's just a complication of that. Anyways, a lot of this stuff we have already talked about, where primase puts down the RNA primer. So know the protein that does that, primase. Then here's your DNA polymerase 3. That's going to start copying it down. Sliding clamp will help like keep the DNA polymerase on the DNA strand. Keep DNA polymerase on strand. Here are your single strand binding proteins. They're going to keep the strand from closing. They keep that replication bubble open. Helicase unzips the DNA strand. And then what they're not showing is what? Think of my cord. What happens as you start unwinding things? They start knotting up. So topoisomerase, the goofy looking one. Okay? So they're not showing that. Anyways, DNA polymerase just gets going. And so here's the next kind of sequence here. This is where the animations, like in Mastering Bio, I know some of you maybe don't actually watch those. You just jump to the questions. Really watch the animations. It can be difficult to fully maybe grasp this with these still frame pictures. Really make sure you watch those animations. Anyways, DNA polymerase will go through and add the new nucleotides. All right, make sure, set everything here. Remember the words anti-parallel. The new strand is anti-parallel, meaning DNA is copied 5 prime to 3 prime, but that parent strand that it's coming from was 3 prime to 5 prime. That's what they mean by the anti-parallel. For the leading strand, only 1 primer is required. As soon as DNA polymerase gets that primer down, it can go. Now getting into the lagging strand, you have to have multiple primers. So that's what they're trying to show here. So here's the same picture I showed you. So we're focusing here on the lagging strand. And there's 2 lagging strands. Okay, 5 prime to 3 prime is here. This is our leading strand. Here was the leading strand on the other side. Lagging strand is because we can only go this way. Like DNA polymerase can only walk this way, but the issue is there's a whole bunch of like things we need to cover back here. So basically DNA polymerase has to like backtrack and do this. Like it's kind of goofy, but that's the only way it can walk down the lagging strand. So, but the mechanics of it are the same idea. You have a primase that puts down a primer for DNA polymerase. DNA polymerase will then go down, copy that, and understand this. So DNA polymerase will make the copy, and then as soon as it hits the primer there, it will then stop, and then DNA polymerase will go back to the next primer that was laid down by primase, and it'll start copying 5 prime to 3 prime until it hits that primer. Then it'll go to the next primer until it's copied the entire lagging strand. So that's what they're trying to show here. So DNA polymerase is going to go until it hits the primer of the next primer. It'll break off and go to the next Okazaki fragment. So that's what they're trying to show on this slide. So DNA polymerase went, hit the primer, breaks off. It then goes to the next primer, copies until it hits the primer it was at before, breaks off, goes to another primer, and so on. Now, the issue is you have these fragments where we have to eventually, and this is RNA, right? So for all of these primers, we need to get rid of the RNA and turn the RNA into DNA. That's what DNA polymerase 1 does. So it's kind of goofy. Polymerase 3, I don't know what 2 is, but polymerase 3 is the main enzyme doing the copying. Polymerase 1 will replace the primers, the RNA primers, with DNA. Okay? And then there's an enzyme called ligase. Ligase is like glue. Ligase will then glue the fragments together. So ligase will glue all of those Okazaki fragments that we made together. Thomas, yeah?
So, when did you Okazaki fragments occur?
So to back up, Okazaki fragments occur on the lagging strand.
So are they just like the ones that like it goes to 5' and then when it goes back, is that the Okazaki fragment?
Yes. Because again, the issue is, if this is the start, DNA polymerase can only go this way. But it has to then copy everything behind it. But DNA polymerase can't just turn around and go 3' to 5'. So that's where the fragments come in. DNA polymerase can only do this. So I don't know another way to describe it. Again, this is where I think watching some of the animations can be helpful. It can be a little hard to picture all this just with some of the still frame photos. So if you have an issue with it, watch some of the animations, and I think it'll help kind of fill in some of those gaps. All right, let me make sure I said everything. So yeah, Okazaki fragment, they happen on the lagging strand. Now the length of the fragments for eukaryotic cells is about 100 to 200 nucleotides long, so really pretty short. They're a bit longer, or actually a lot longer. And E. coli cells, you don't need to memorize that. It's just to give you an idea of the scale you're working with. Questions so far? And then again, DNA ligase will then put them all together. This slide brings it all together, theoretically. So here's our origin of replication. Again, the leading strand, this is 5', that's 3', 5', 3'. So here they're just focusing on 1/2 of that bubble. So DNA polymerase, that's 5', that's 3'. We got no issue DNA polymerase doing 5' to 3'. The issue is the other strand, DNA polymerase can only go this way. So this is where the Okazaki fragments come in. DNA polymerase, here's the primer laid down. It'll go until it hits the primer of the next strand. It'll then break off. So DNA polymerase hits that last Okazaki fragment, and it loops back to where the helicase opens up the next bit of DNA, and primase put down the primer, and then DNA polymerase can start going until it hits the primer of the Okazaki fragment it had originally done. It'll just keep doing this. So it'll look like that until it's copied the entire lagging strand. Okay? DNA polymerase 1, the first DNA polymerase, its job is to replace RNA with DNA. So replaces RNA to DNA. Ligase, that's the glue. It's going to glue the fragments together. These are the single-strand binding proteins that keeps the DNA from going back together. Topoisomerase prevents the DNA from kinking up as helicase is opening it. All right, just spend some time processing it. Now, as I've been showing you, on this last slide, on Where'd it go? This slide. Basically, how the vibe I was kind of showing you was that the DNA is stationary and the proteins are moving to copy the DNA. Now, an alternative model of DNA replication is called the trombone model. The trombone model says that the DNA replication complex, so the DNA replication complex is all of the proteins. So all those proteins, the polymerase, the helicase, single-strand binding protein, all of those, they all make up the complex. The idea of the trombone model is the complex they're saying is, hey, what if the complex is stationary? The idea is that the complex would be anchored to the nuclear envelope. So all of those proteins are anchored to the nuclear envelope. Think of it like a car wash. Where what's up? Think of it in the trombone model. Think of the DNA replication complex, all those proteins, as like a car wash, and the DNA would be like the cars that will move through the car wash and get cleaned. Or in this case, they get their new nucleotides put down. That's the idea behind the trombone model. That's really about all you need to know. I doubt you're gonna have an essay question where they're gonna have you like actually describe the model, but why they call it the trombone model is from the lagging strand. So the lagging strand, it basically like as helicase opens up the DNA strand, the lagging strand will start to wrap behind itself, and then the idea would be for both of the strands that DNA polymerase, like the car wash, is pulling the DNA through and adding the new nucleotides. So like here, DNA polymerase would essentially be pulling all these nucleotides, this DNA strand towards itself, adding the new nucleotides, and then once it's done, it'll then relax and then pull in the next strand of DNA. If that's a little vague for you, I'm trying to picture it. It's not a huge deal, but the main idea is that why this model exists is just getting at this idea that maybe the replication complex isn't moving at all, it's just anchored in the nuclear envelope. That's the point of the trombone model. Okay? So there you go. All right, so getting into some of the errors, like how accurate is this? It's actually pretty accurate. There's only, when you're copying DNA, 1 in every 10 billion added nucleotides is there an error. So appreciate how accurate that is. Like, that's really, really accurate. There are systems, like if, say there is an error, there's systems in place. There are other DNA polymerases that can proofread the errors. And like, let's say you put a C where there should have been a T. There's a DNA polymerase that can remove the error and replace it with the right nucleotide. And we call that mismatch repair. So enzymes that can then repair any mismatches. And this is people who have like, there are certain people who have like, they inherit like faulty repair enzyme genes. And so they found this in like certain colon cancers. So people who inherit a faulty enzyme, DNA repair enzyme, they're more at risk of colon cancer where, you know, colon, like those cells there are constantly being turned over because of how like acidic the environment can be, chemicals that might be in food. And so if you don't have very good polymerases that can repair the mismatch and damaged DNA, that's going to increase your chances that you're going to get a cancerous cell. So the idea would be slowly but surely errors can creep in. Not very often, errors can creep in over all of our lifetimes. And as like every time you copy a cell, you copy all that DNA, you might introduce like a few different errors. So, you know, over the course of our entire lifespan, those few errors can lead up to decent amounts of errors, which can slowly increase your risk that you'll get one of those cells that becomes a cancer cell. That's the idea of like errors and mutations in cancer. Okay. All right. So looking at like how exactly like we would go through and fix errors, This is focusing on what's called a thymine dimer. So let's say spring break 2023, right? I'm going to sit on the beach and just get sunburned. Well, what can happen is if you get exposed to too much UV radiation from the sun, if you have two thymines, so you have two T's next to each other, the UV radiation can cause them to get glued together, get stuck together. The issue with that is whenever the cell's trying to then copy that DNA, that will basically gum up the DNA polymerase machine and increase the likelihood that there's an error that happens. Well, fortunately, we have these things called nucleases. So nucleases, think of them as scissors. They're like genetic scissors. Nuclease will come in and it'll cut the DNA strand there, and then DNA polymerase comes in and it'll fill in the proper DNA base pair, and then ligase, that's the glue, ligase will then glue those two strands back together. Okay? Nuclease, scissor, polymerase adds in the bases, then ligase glues the strands back together. So actually a fairly simple mechanism, kind of intuitive. All right. That's really most of it. Let me make sure I said everything here. Other things that can damage DNA be like X-rays, certain chemicals, especially a lot of processed meats. They can have some chemicals that can cause DNA damage. So again, that error rate improved. So very low error rate for adding nucleotides, and also a very low error rate in proofreading. So our body is really good at finding those errors, but it still every so often can happen where we add in the wrong nucleotide and we don't catch the error, and then we get a mutation, a mismatched DNA put in place that we won't catch on. Now, thinking back to just thinking of evolution, mutations, that is the main source of natural selection. Is anything that you remember about evolution is mutations are the main source of variation. So most mutations are not helpful. Most of the time you get a mutation, and that is not going to help the cell. It's going to usually cause a bad protein or a protein that doesn't work as well. But every so often we can make a mutation, a change in that DNA sequence, and that might lead to a more helpful enzyme or more helpful protein. Might make the giraffe's neck slightly longer or something. Then natural selection can select for that trait. We'll talk about that in the next unit when we go over evolution and stuff, but just keep that in mind. Okay, so this is really interesting. So talking about living forever, you ever wonder that what prevents ourselves from kind of living forever? Well, there's a lot of different things, but one key thing is whenever we copy the DNA in our cells, we can't copy all the way to the end of the chromosome. Because DNA polymerase, we can't complete that final 5' end. We end up having a very short sequence of DNA that does not get copied, so it then gets cut off. That end of DNA that doesn't get copied, we call those telomeres. So the body knows this. It knows it's not going to copy all the DNA, so it has basically a bunch of random, they call them special nucleotide sequences. They're non-coding sequences, meaning they don't actually code for proteins. They're not important sequences. They're just ends that will get cut off because we can't copy the ends of chromosomes. It's like a buffer zone essentially for the DNA. Anyways, here are the telomeres shown on the chromosomes. So the issue is each time we do DNA replication, we will cut off a little bit of those telomeres. We do another round of DNA replication, we'll cut off another part of the telomere. So then what's the issue? Each time you cut off a little bit of the telomere, you eventually run out, right? And so then you're like, okay, well, we ran out of telomeres. Well, then we start cutting off actual coding portions of the DNA, actual regions of the DNA that make important proteins. So let's say you then cut off this portion of you remove this gene that controls the cell cycle. Well, then shoot, now you're increasing the risk of cancer. That's the idea of why there's kind of a limit on our cells. Now, a lot of like, especially plant cells, are really good at actually repairing their telomeres. So a lot of research in like extending human life is how can we prevent the telomeres from shortening. So we do know that there are enzymes called telomerase. Is that on this slide? Yeah, I'll go back to that last slide. But telomerase, that's an enzyme that will lengthen the telomeres. So we actually have telomerase in our germ cells. Our germ cells make the gametes. And, we want to keep our species going, right? We want to make sure we can make enough sperm and eggs. So we don't want to keep these germ cells, they have to do a lot of meiosis. So their telomeres are under a lot of like pressure, a lot of shortening. So our germ cells, they actually have the enzyme that can keep extending that telomere. But none of our other cells have it. Like your skin cells don't have telomerase, but your other cells do. So basically, extending life research would be like, how can we essentially like activate or add in telomerase to some of our other cells to prevent those telomeres from shortening. So it's kind of a very interesting idea. Let me make sure I said everything on this slide. Okay, again, telomeres don't contain genes, just these repetitions of short sequences. And yeah, the telomeres, the DNA does keep getting shorter after each round of replication. It's just the telomeres prevent you from like removing critical genes. Well, it doesn't really prevent you. It just postpones removing the critical genes. All right, questions? Any of that? Okay, that's all I got. So